So today I will present you in about 20 minutes one of the projects that we have been working on over the past a couple of years. Towards the very end, I will also have one summary slide where I just outline some of the other interests that we have in our group. Okay, so to me, one of the most fascinating or striking features of biological systems is their ability to create very, very complex form and function in a highly robust and predictable manner. And so this is even more fascinating when we realize that many of the underlying physicochemical processes are substantially stochastic. And so this is because on small length scales, such as the scale of a cell or an organelle, random thermal fluctuations can become very significant, as we heard also before. And so we know by now that stochasticity in chemical reactions can very strongly affect the molecular constitution and physiology of genetically identical cells. But at the same time, networks of chemical reactions coordinate highly complex spatiotemporal programs, for example, when a cell develops into a fully formed multicellular organism. And so how can we reconcile the remarkable robustness and predictability of biological systems with a very large degree of stochasticity that we observe at smaller scales? So to perhaps paraphrase this question, how can highly predictable outcomes emerge from hardly predictable building blocks? And to give you a slightly naive analogy here, we do understand nowadays how complex and robust functionality can be created from smaller building blocks. And we can do so because we have physical laws that precisely predict the behavior of individual components, but also the relevant emergent behaviors when thousands or even millions of such components interact with one another. So now biological systems have to deal with a substantially more challenging situation because the physical processes that are available to them are often substantially stochastic, which is a little bit like trying to build an Airbus from unreliable components. And so we want to understand how biological systems solve this problem and what strategies they can use to control, buffer, even utilize stochasticity to ensure reliable outcomes. And to do so, my group merges concepts from stochastic processes, control theory, and statistical physics, and we apply these concepts to different biological systems in collaboration with our experimental partners. And today, I want to give you one concrete example of this, where we've studied how the formation of mesoscale subcellular compartments can provide robustness to biochemical systems. Okay, so I've mentioned to you before that stochasticity in chemical reactions can lead to large fluctuations in cellular behaviors, and the probably best studied example of this is gene expression, where random events in both transcription and translation can lead to large fluctuations in gene expression output. And we can see this, for example, very nicely when we measure transcription in living cells, where we can see mRNA transcripts as they are made at the gene locus, here in the case of a breast cancer cell. And so from this movie, you can appreciate the very bursty behavior of transcription, where random pulses of transcriptional activity are followed by long silent periods. And as a consequence of this, both mRNA and protein concentrations can vary substantially over time, but also between genetically identical cells. And so a central question in the field has been how cells can sort of safeguard their biochemical programs against random fluctuations that arise in gene expression. And so one potential strategy to achieve this are negative feedback circuits, such as, for example, here a protein inhibiting or regulating its own transcription. However, we know from theory that in cells, such feedback mechanisms can be very strongly limited due to certain kinetic constraints, and that being precise through feedback control would be extremely costly for the cell. And so this is in fact supported if we look at system-wide single-cell studies that measure noise, so protein concentration noise, across the whole proteome. And so here, noise is defined as the standard deviation over the mean. So it gives you basically a relative variation, a percent variation of expression level relative to the mean. And so here, each circle here is one protein species, and what you can see here is that noise initially decreases for increasing expression levels, but then saturates at a certain plateau from which it normally does not increase any further. And usually this plateau is somewhere in the range of 20 to 40% relative variation, depending on the organism and condition that we are looking at. And so these studies suggest that, in fact, gene expression is universally noisy across the proteome, and that the precision that cells can achieve through genetic circuits to control protein concentration is in fact, very limited. And so more recently, people started thinking about what other potential mechanisms cells could use to ensure more precise behaviors when precision really matters. And so one hypothesis that came up more recently is that the spatial organization of molecules into phase-separated membraneless organelles could provide such a mechanism. And so this hypothesis is based on the very characteristic thermodynamic properties of phase-separating systems, which as soon as the total concentration of a certain phase-separating protein exhibits a certain threshold concentration, demixes into two distinct phases, one a dilute phase with lower concentration, and then a condensed droplet phase with higher concentration. And importantly, the concentrations in each of those two phases are thermodynamically constrained, as can be described, for example, by a phase diagram. And so now, consequently, if the total concentration in the system changes, we would expect the phase-separated compartment to change its size until the outside concentration goes back to its preferred equilibrium concentration. And so this idea suggests that phase-separated condensates could serve as sort of dynamic storage compartments which buffer fluctuations in total concentration, or in other words, they could absorb fluctuations in the total concentration by dynamically changing their size. Now, however, whether this ideal and very simple thermodynamic picture really holds inside the very complex environment of a cell is not immediately clear. And so, for instance, this idea is based on the equilibrium physics of macroscopic phase-separating systems, where cellular systems tend to be rather small and generally not at equilibrium, for instance, because material is actively produced and degraded. And moreover, of course, cellular compartments can be very complex and rich in the components that they involve, which of course may further complicate this simple picture. Okay, and so to test if and under what conditions phase-separated compartments can constrain cellular noise, we first used a theoretical approach and subsequently performed experiments in single cells to test these theoretical findings experimentally. Before I proceed, I just want to acknowledge my collaborators on this project, and in particular, Adam Klosin and Florian Olch, who really did most of the work that I'll be showing to you. Okay, so on the theory side, the key challenge was to come up with a theoretical framework that bridges between the stochastic kinetics of protein expression and the mesoscale organization of molecules into phase-separated compartments. And so on the next couple of slides, I will first discuss these two parts in isolation and then show how we can bring them together. Okay, so to describe protein concentration noise, we consider a very, very simple kinetic model of a constitutively expressed gene, where RNA and protein molecules are stochastically produced and degraded with constant rate parameters K1 to K4. And then next to intrinsic fluctuations in transcription and translation, we know that protein expression is often very significantly affected by so-called extrinsic variability, which stems from cell-to-cell differences in the cellular microenvironment, such as ATP abundances, ribosomal abundances, cell cycle stage, and so forth. And so here we account for extrinsic variability by considering the transcription rate K1 to be randomly distributed across cells. It's not fixed, but it has a distribution across the population, which could, for example, serve as a proxy for plasmid copy variation. And so in summary, this gives us a very, very simple stochastic model, which accounts for both intrinsic and extrinsic variations in gene expression. And now, importantly, if we look at the steady-state noise of this system, then it captures the characteristic inverse scaling of noise with average concentration that I've shown you before, where noise first decreases with average concentration, but then plateaus off. And so just to mention here, the idea here is not to be biologically precise, but really just to have a very, very simple model that captures the statistics and the scaling of noise with average concentration. Okay, and as a next step, we will for a moment forget about the protein expression dynamics and focus exclusively on the process of phase separation. And to describe phase separation, we again focus on a very simple thermodynamic model of a binary mixture where a protein here indicated by the red circles can partition into a droplet. And so here N plus and N minus are the number of proteins outside and inside the compartment, and N, which is just the sum of the two, is the total number of proteins that I have in my system, and which for the moment I consider to be fixed. Okay? So here are just a couple of more details. So we consider spherical droplets, which are homogeneous, and we consider that they're surrounded by a dilute phase with lower concentration, which is also homogeneous. And moreover, since we focus on fluctuations in the dilute phase, so the outside of the compartments, we neglect density fluctuations inside of the compartment because those are expected to have very little influence on dilute phase concentration. And we can then introduce corresponding volume fractions, which are really just concentrations multiplied by the molecular volume, so we can really think of them as normalized concentrations. And so phi here is the total concentration, and phi plus is the concentration in the dilute phase. And we can then describe the system using a free energy. So I apologize, I don't show the explicit expression here because it's a little bit bulky, but in essence consists of three parts. One part corresponding to the droplet, one part corresponding to the dilute phase, and one part that comes from establishing the interface between the two phases, which is essentially a surface tension times a surface area. Okay, and we now want to take this simple equilibrium model and extend it to the case where the protein concentration can now fluctuate, as we have described previously using our kinetic theory. And to do so, we go back to this kinetic model and introduce two additional exchange events or exchange reactions with rates k in and k out, which allow the protein to move between the two phases once they have been made. And so importantly, we want to choose these rates such that they are consistent with the thermodynamics of phase separation. And to do so, we consider the rates to satisfy local detail balance such that the ratio of the forward and backward rates is just given by the corresponding free energy difference as long as the total amount of protein remains fixed. And if you now fix one of the two rates, in this case, for example, by considering the in rate, so molecules moving into the droplet to be given by diffusion, then we also know the corresponding reverse rate. And so now, as long as the total concentration remains fixed, the system exhibits detailed balance, we will converge to our equilibrium distribution. But as soon as I switch on the dynamics of protein expression, a detailed balance will be broken, and I'm driving away the system from equilibrium. And I can now describe the joint non-equilibrium dynamics of the system using a master equation. So we have a kinetic description of the system that captures the statistics of the total protein concentration phi together with the concentration outside of the compartment, so the dilute phase concentration. And so again, this master equation is a little bit bulky, but in essence consists of two major contributions. One that comes from the kinetics of protein expression, so how RNase proteins are made and degraded, and then one part which conserves mass, which is responsible for partitioning the proteins between the two phases. All right, and from this master equation, I can then calculate arbitrary statistics of dilute and total concentration. And so that now really gives me a way to compare how much noise is there in the total concentration and how much noise remains in the dilute phase concentration once compartments form. Okay, so on this slide, I want to summarize the most important results or predictions that came out of this analysis. And so this plot shows the predicted noise, again defined as a coefficient of variation, standard deviation by mean, in the total and the dilute phase. And so we can compare them to each other. And we do that for increasing mean expression levels. And so we see that for undersaturated systems, so that's when the system doesn't have sufficiently large concentration to form compartments, we see again this characteristic inverse scaling of noise with average concentration, where we then reach a certain plateau from which normally, and normally means in the absence of compartments, it would not decrease further. But in a phase-separating system, as soon as we approach a certain threshold concentration, compartments begin to form, which then leads to a quite strong reduction of concentration variability down to a certain plateau. Using a concept from probability theory known as the law of total variance, one can then show that within this limit, so within this plateau, essentially all the remaining noise is due to the thermal fluctuations in phase separation. So while noise that comes from protein expression is suppressed almost entirely. And so in other words, we are basically trading noise in protein expression for noise that is introduced by phase separation. So that's the price one has to pay to make molecules form form a compartment. But since phase separation noise is typically very small for systems of realistic size, it's typically a relatively good deal for the cell, as shown here also in this plot.
So Christoph, on the x-axis, is that phi or phi plus?
This is phi, so phi total. Phi total. You can do the same plot for phi plus, or have two x-axes, which is a little bit weird to look at. But this is here on the x-axis is definitely phi total.
Right, but so phi plus also goes down, right?
Phi plus stays at a certain point then, right? So it would actually look, so the curve would stop somewhere over there. If you know what I mean, because the system is saturating and the average concentration will not just continue growing. Yeah, okay, that's good. Okay, so this the green curve here corresponds to the case where phase separation is very fast on the timescale of protein turnover. However, we know from proteomic studies that protein synthesis rates and degradation rates can range over orders of magnitude, which makes it unclear whether such a strict timescale separation can always be expected inside the cell. We therefore wanted to know what happens or how this behavior changes when the timescales of phase separation and protein turnover become comparable. And to do so, we introduced two timescales here. So the diffusion time tau d, so that's the average time it takes for a protein to diffuse into the compartment, and the average protein lifetime tau p. And we see as soon as the timescales are no longer distinct, the reduction of noise becomes less pronounced, and interestingly also has a certain qualitative difference because it now has a minimum, so there's an optimal expression level for which the noise is lowest, but then increases again. And the reason for that is that for large expression levels, so the reason why this curve goes back up is that for large expression levels, protein synthesis becomes fast in comparison to the diffusion of protein into the droplet, such that they can sort of pile up in the dilute phase, in which case noise can be constrained less effectively. And so in summary, this theoretical model predicts that concentration noise should be effectively reduced by phase separation as long as phase separation is comparably fast on the timescale of protein expression. Okay, and so we wanted to test whether that's the case inside cells, and so we therefore teamed up with the lab of Tony Hyman, and in particular Adam Closine, who measured all the data that you will see. And in particular, we studied a variant of a protein called DDX4, which forms liquid-like compartments in the nucleus of mammalian cells. And using transient transfection, we could then measure concentration noise in that system for a very broad range of expression levels. And so this data show that concentration noise was in fact strongly reduced as soon as compartments started to form, and moreover it also shows this minimum of noise at a certain point, which was predicted also by our non-equilibrium theory. And if you compare these measurements now with our model, then the two curves are in, I would say, relatively good qualitative agreement, whereas the model predicts slightly less noise than what we see in the data, and this may of course stem from the fact that there may be additional sources of variability in the experimental system, which are not captured by our very minimalistic model. Okay, so here's just one additional movie that I want to show you, which I believe illustrates the reduction of noise by phase separation quite well. And so what we did here is that we made use of the fact that many membraneless organelles dissolve during mitosis, so we could actually follow the cells through mitosis, see what happens when the compartments go away, and then compare noise levels in the presence or absence of compartments. And so this is what you see here now in this movie. You see that this is now an interface, but then where we see a relatively narrow distribution. You can see the scatter here is relatively narrow if you measure concentrations outside of the compartments. But as soon as the cells enter mitosis, the cells round up nicely, and you can now see a very large degree of variability sort of unleashed as soon as the cells exit mitosis, go into interphase, compartments form again very quickly, and you can see again this reduced noise sort of state. Okay, so if we now take a step back, so the system or the data that I've shown you here are based on an engineered condensate where a single phase separating protein partitions into a compartment or into droplets. And now, of course, endogenous compartments can be much more complex than this, as I've mentioned also before, very often involving many different types of molecules and interactions, and additional activities such as post-translational modifications and so forth. And so the question is then, of course, how far can these relatively simple concepts take us inside living cells? And so we therefore wanted to test this idea also an endogenous multi-component condensate at physiological concentration. And in particular, we quantified endogenously labeled NPM1, so nucleophosmin, so that's a key component of the nucleolus. The nucleolus is a large multi-component compartment with very complex biochemistry. And we then did the same trick as before. So we measured concentration noise during mitosis, so that's when the nucleolus is absent, and compared it to interphase when NPM1 partitions into the nucleolus. And so also in this case, we found that nucleoplasmic NPM1 concentration is strongly reduced. If you now look at the scatter plot that shows total concentration or total intensity in this case versus dilute phase intensity, and we measure the variation along the x and the y-axis, which is seen on the plot here on the right-hand side. And then you see that the second distribution is squeezed together. And so also in this case, noise in the dilute phase is very strongly constrained in the presence of nucleoli. And in summary, these experiments demonstrate that noise can be constrained very effectively also by an endogenous multi-component condensate at physiological concentration. Okay, so I want to conclude here. So I've shown you that cellular noise is effectively constrained by phase-separated compartments. And so this happens because condensates serve as sort of dynamic buffers that want to maintain outside concentrations within narrow ranges. I've also shown you that the extent to which that occurs depends critically on the timescales involved in protein expression and phase separation, such as diffusion constants, protein synthesis, degradation rates. That also highlights the importance of using sort of non-equilibrium approaches that can capture the interplay between such timescales. And finally, I want to say that, of course, everything I've shown you here is really just a starting point, and there are many open questions that need to be addressed in the future. And so, for example, in our theory, as you've seen before, we have so far focused on systems that consist of a single phase separating component, whereas, as I've shown you before also, cellular condensates can be much more complex, involving many different types of molecules and additional chemical activity. And we are currently working on a more general Langevin formalism that captures fluctuations in such systems. In this context, I also want to mention some recent work from the Safran group, who have addressed also the first point here about multiple components to understand how noise can be buffered by multi-component phase separation, for which the thermodynamic constraints and phase diagrams can be more complex. So now on a more conceptual level, a key question that also remains is, of course, how general this concept is in the context of endogenous condensates, and in particular, to what extent cells really use this to control downstream chemical processes which partition into compartments. And so based on our previous work, we speculate that compartmentalized proteins, but maybe also RNAs, could exhibit substantially less noise than those that do not localize with compartments, and therefore provide a more general way to maintain precision despite high levels of gene expression noise. And if that was indeed true, it would have very interesting implications for intracellular control and information processing. Okay, so as I promised, I'm running out of time actually, but I just wanted to show you a few more other things that I'm working on. I will not go into detail of those, but we are generally interested in phenomena where stochasticity plays an important role. I just want to highlight one of them, which is the one up there, and I want to highlight it because I think it would also fit relatively nicely into the theme of this conference, into a different session probably, which is essentially a collaboration between the Hansen lab at MIT and Leonid Mini's lab at MIT, where we used super resolution macroscopy in combination with statistically inverse approaches in polymer physics to understand the dynamics of chromatin looping inside living cells. So if you happen to be interested in this, please chat to me. I would be excited to talk about this with you. Okay, I want to stop here. I want to finally thank my collaborators on this project, in particular Adam and Florian, who really did most of the work that I've shown to you. I also want to thank the rest of my wonderful lab and all my other collaborators, and of course you for your attention.